Tuberculosis Tales, Mycobacterium's Whispered Trails. I am the cause of tuberculosis, TB, which the World Health Organization, WHO, classified as a deadly respiratory disease after I threatened the world in 1993. In fact, I'm so famous that the WHO publishes epidemic reports all about me every year. The 2019 report estimated that 10 million people were infected and around 1.4 million people suffered mortality. According to some models, the COVID-19 epidemic may worsen TB burden due to undernutrition, multi-drug resistant TB strains, unemployment, and redirection of resources to fight COVID-19. Who am I? Mycobacterium tuberculosis. I am a gram-positive, acid-fast bacillus with a very tough cell wall composed of lipopolysaccharide, mycolic acids, arabinogalactan, a polymer of arabinose and galactose, a thick peptidoglycan layer, and a lipid bilayer. Over 60% of my cell wall is made of mycolic acids, which contribute to my virulence. Other lipids in my cell wall, such as sulfolipids, cholesterol, and liporabinomannine, also help to increase my virulence. They help me be resistant to antibiotics, resistant to osmotic lysis, and oxidative stress. I hail from the bacteria kingdom, Actinobacteria phylum and class, Actinomycetales order, Mycobacteriaceae family, Mycobacterium genus, and tuberculosis species. I have several other relatives that form what is called the tuberculosis complex, because we all cause tuberculosis disease in either humans or animals. They are M. africanum, M. bovis, M. mungi, M. canneti, M. orygus, M. microti, M. caprae, and M. pinnipedi. One of my relatives causes leprosy, and their name is Mycobacterium lepra. Not all of us cause tuberculosis, though, such as Mycobacterium smegmatis and Mycobacterium fortuitum, but they can cause human infections. Humans are my only known host, and I am spread primarily through respiratory droplets, such as sneezing or coughing. I am famous for invading the alveolar sacs of the lungs, but also invade the lymph nodes, bones, joints, central nervous system, heart pericardium, or bloodstream. I cause humans to cough, have chest pain, lose weight, fever, and even night sweats. If in the lymph nodes, the person can get abscesses. If in the central nervous system, I get quite a headache. My metabolism is heavily dependent on lipids, which are my primary carbon and energy sources. I'm such a chemoheterotroph. I steal lipids from my host because they are delicious, but I can also make my own. In fact, I can use cholesterol as my sole carbon source. Important enzymes responsible for fatty acid biosynthesis are FAS1 and FAS2, which produce some mycolic acids that make up part of my cell wall, such as PDIM, sulfolipids, polyacyl trehalose, and diacyl trehalose. Scientists predict that under aerobic and early growth conditions, I use glucose and triglycerides as carbon and energy sources, but then switch to using lipids once inside host macrophages where I cannot access glucose. Take that, macrophages, I can still replicate. Sometimes, a bunch of macrophages will chase me down and form granulomas by clustering together in large groups of foamy cells which starve me of oxygen. I must change up my metabolism if I want to survive, so I alter my Krebs cycle to favor succinate production. Succinate feeds succinate dehydrogenase, which is involved in both the Krebs cycle and electron transport chain. This means I still get to make ATP. To increase the success of my invasion, I must reduce the population of my enemies. Studies in TB patients have shown that I am associated with a decrease in microbiome diversity, especially short-chain fatty acid or SCFA-producing bacteria such as firmicutes and Bacteroides. You see, SCFAs, such as butyrate, are helpful in immunomodulation, meaning that they help stimulate lymphocytes and phagocytes. I don't want that. Gut and lung dysbioses are my friends. I don't dislike all microbes in the human microbiome, though. I do like proteobacteria, so they are overrepresented during MTB infection. During anaerobic growth, they produce mixed acids, including succinate, lactate, formate, but also dreaded SCFAs. However, as I mentioned before, I do like succinate for my own ATP production. I also like some actinobacteria, such as from the genus Collinsella. No one knows exactly why these groups are overrepresented, and I'm not telling. I have many clever ways to escape the human immune system. One way is by purposely allowing myself to be internalized by alveolar macrophages, but then preventing the maturation of the phagosome. You see, during normal phagocytosis, the phagosome fuses with the lysosome to form a phagolysosome, which contains an acidic environment and enzymes that can kill me. One way I prevent lysosomes from killing me is by secreting a lysosomotropic, can penetrate lysosomes lipid called 1-TBAD, which functions as an antacid. Another way I do it is by secreting a protein, tyrosine phosphatase, PTPA, which prevents VATPase in macrophages from doing its job of actively pumping protons and acidifying the phagosomal environment. Since I can damage phagosomes with my ESX1 effector proteins, macrophages try to come and repair them via the endosomal sorting complex required for transport, or ESCRT, and Gal3 proteins. However, my handy effectors ESXH and ESXG interfere with the recruitment of ESCRT. This prevents maturation into a phagolysosome and thus keeps the pH high enough to keep me comfy. Humans have found that ESXH and ESXG complex with each other and prevent ESCRT recruitment, thus disrupting its function, which also includes moving me to lysosomes. Gal3 gets recruited to damaged phagosomes when internal glycoproteins get exposed, but my effectors block that signaling too.